Throughout history, humankind has endeavored to create the greatest works of art and testament to the human spirit and ingenuity. Rodin, Da Vinci, the Greek classics, stick figure Bob, but above all else stands the greatest work of art and science to outshine them all, the four modes of transcriptional regulation. But exactly what are we looking at? Why is this so important? Well, transcriptional regulation is an essential method for determining when and how much a particular gene is transcribed into RNA in prokaryotic organisms. Without such regulations, genes might constantly be turned on or off with lethal effect. There are several actors in prokaryotic transcriptional regulation: the regulator gene, promoter, operator, structural genes, and RNA polymerase. The default activity of transcription for a gene or set of genes can be identified by the sequence of the promoter. Likewise, within the promoter, consensus sequences can be found at the -35 and -10 region upstream from the transcription start site. These consensus sequences allow signal factor to bind, guiding RNA polymerase to bind onto the promoter region to begin transcription. However, in order to regulate transcription, a regulator gene also produces a repressor or activator protein that can repress or activate RNA polymerase activity. These proteins can be active at all times or inactive at all times until influenced by a small effector molecule. These multiple combinations of active and inactive repressor and activator proteins give rise to the beauty of art, the four modes of transcriptional regulation. The four modes are negative inducible, positive inducible, negative repressible, and positive repressible. Today we shall take a closer look at positive repressible. In positive repressible, the regulatory gene is an active activator gene. This means that it codes for a protein that, when bound to the operator site, allows for transcription to occur continuously. The operator site is simply a designated sequence that overlaps with the promoter and structural gene region, allowing for the active activator protein to bind. Downstream from the promoter region are the structural genes. These structural genes are responsible for coding the RNA sequences. There can be several structural genes here, or just one, depending on the genome of the organism. So say if we wanted to code for proteins that give a bacterial cell a blue color and enzymes that can process lactose at nearly all times, then we would find those genes here. However, a small effector molecule can interfere with transcription by binding to the active activator protein. By binding, it inactivates the activator, causing it to be dissociated from the operator site. Without this, the RNA polymerase will no longer bind to the promoter region, effectively ending transcription. A great analogy for positive repressible is making tea. I like strongly flavored loose leaf black tea. As long as I leave my black tea diffuser in my teacup, the tea leaves will continue to diffuse, making stronger tea. However, at some point, my cat comes along and I have a higher affinity for petting my cat than tending to my tea. At this point, no more black tea is being diffused. So the regulatory gene product here is me, and the small effector molecule is my cat.